In this video, I want to do a quick introduction to Lab 19. The formatting here looks a little different than your lab manual. It's fine, it's the same content. So as you can see, right, acids, bases, pH, and buffers. Let's take a look. Most of this information is talked about in the lecture videos. Background about pH, how to calculate pH, what buffers are, how they work. Notice Timberlake is giving you some example calculations here and some background information about each part. It's worth reading through. I think it's also useful to read through the experimental part just so, you're going to observe the experiment, but take a few minutes just to get a little familiar with what's going to happen in the videos. Here and there, I have changed the procedure slightly, probably a little bit different volumes, etc. Observation should be fine for you to be able to record and fill out your report sheet. For this lab, we will want to complete the pre-lab questions. And just like all our labs, if we complete a part of the lab through a video or in the lab, then what we'll want to do is then answer any questions, any follow-up questions with that particular part. Part A doesn't have any follow-up questions, so let's just take a quick peek. And actually in the video, I talk about this as well. As it turns out, simple red cabbage juice can be used as a pH indicator. And what I mean by pH indicator is that this thing is going to change color based on pH. Now, you'll see in the video, I'll mention it in the video, we're not using a complete set of buffers here. We're just using the odd number buffers. And that gives you plenty of range in the pH values and the pH colors. Very simple reaction. I add a little buffer to a test tube, I add a little bit of the red cabbage juice to the test tube, and you see a color change. And what you'll want to do is write down the color that you see in these different boxes. The final image of all these different colors are going to be posted with this material on Moodle, because these colors are then going to be used in your other parts. So this is part B. Again, the formatting a little different than yours. In the part B video, what you're going to see first is this part. And I'm not using a pH meter. Instead, we're using pH paper here. It has indicators within the paper. And so when you place a little bit of vinegar on the pH paper, it turns a red color, indicating the pH of the solution. It's like a test strip. And then in the video, you'll match a color of the chart to figure out the pH, approximate pH of all these solutions. And then the second part that I do is here, and indicator, well, that's the red cabbage juice. So now each one is going to have a color. Right, so you'll record the color here. And then you're going to use the part A colors, or the colors in part A, to then figure out the approximate pH here. Right, because you're going to have all these different colors written down. The image will be available to you as well. And then you can approximate the pH here. If the color looks something in between the colors for 3 and 5, then it's fine to write down pH of 4. Use your own judgment. Most of these items are on the video. If there's not the item, then just replace it with the item that's given. For example, I think instead of apple juice, we have grape juice. So just substitute it there. So we complete part B, and then we'll want to go in and then write down answer these questions all the way down. So basically you're completing the entire lab, and I'll make a note on this at the top. And again, don't worry, the notation here is just bad the way I had to convert it to be able to do the notes on it, actually. Last part, so we're testing buffers. And remember what a buffer does. It's just a solution that resists pH changes. pH changes when, an acid or a base is added to the solution. Solutions are the same for each one, right? This set we're going to add acid, this set we're going to add a base. You'll see in the video, you'll use the color of the solution changes, because I'm adding red cabbage juice here, to then approximate the pH before and after the acid and the base are added. The high pH buffer has a pH equal to 10, and the low pH buffer has a pH of 4. And I talk about this experiment as I do it. And, just like all the other labs, if we do a particular part, then we're going to complete the questions associated with that part. And that's it. So you'll complete the entire lab.